All right, everybody, let's talk about osmotic pressure. And again, this is one of those growth condition labs where we're evaluating what it takes for these microorganisms to survive in an ever-changing environment and the mechanisms that they use in order to survive. So osmotic pressure, well, what are we thinking of? What are we talking about? What word does this remind us of? What are we thinking about here? Osmotic pressure. What comes to mind? Well, possibly it is osmosis. Okay, and what is osmosis? Maybe you've heard that before, maybe not. Think about that for a sec. Osmosis, osmotic pressure. What's moving here? Well, what is moving here is, let's see if I can erase it all, water. H2O, yay, it's water. Let's actually just put it in as water here. Okay, so we're talking about the movement of water. Now, how does it move? Does this take energy? Does this not take energy? What do you think? What's going on? Is water just one of those things that can freely kind of move around wherever, or is it something that needs help? Well, think about it. What does water look like, right? We got that big old oxygen and these two little hydrogens. It's got some, areas. It's a polar molecule. All kinds of fun stuff. Very electronegative with that oxygen. But yeah, it just kind of passes through everything. Passes through membranes freely. Does all kinds of stuff. You can hear, if you go to the grocery store, especially now everybody's dealing with bottled water and stuff like that. Some water like Aquafina and Dasani, they're purified. It's just tap water basically, purified through reverse osmosis. So they just take basically some type of apparatus and they put a membrane there. And they basically force the water through the membrane. So the fresh water is over here, and all the solutes, or the stuff that we want to get rid of, salt, sugars, oils, whatever, can't pass through that membrane. And so here's your fresh water over there. Well, anyways, not to get distracted. But that's what we are talking about. We're talking about movement of water, and it's passive movement. And well, how does it work? Well, it goes against a concentration gradient. So if you got water here, let's say we have this kind of tube thing. Let's say there's lots of, this is going to be a terrible drawing, I can already see. Lots of stuff here, not so much stuff here. The water's going to want to go against the gradient. It wants to dilute things. And you can think of it like, that was a terrible diagram, I apologize. You can think of it like your own cells, right? Our cells, they've got stuff in them, right? They're ignoring organelles and stuff like that, but there is stuff inside of our cells. If you drop that cell into, let's say, like a bowl of salt water, right? Maybe there's a higher concentration of salt in the bowl outside of the cell. So what happens? Well, that water that's in there, it wants to rush out. It wants to dilute everything. It wants to find balance. It wants to make everything the same, but the water is going to rush out. Not the solutes, not the salt, not the sugars. Those are stuck. They can't go inside, outside. Now, yes, our cells do have these pumps that move things in and out, right? Sodium pumps, calcium pumps, yes. Bacteria have the same thing, but really water can freely move in and out. There's also, we have special valves and stuff like that can flush more water out and stuff like that too, because water is very important for the stress of things. Okay, but it's all about the movement of water. And it has to do with how it interacts and in relationship to the environment. So there's really three situations we want to think about. So here we've got a cell. Right, let's put a cell here, put a cell here, put a cell here. Okay, so for this first cell, and remember they have this cell wall, because we're talking about bacteria. Not all bacteria have cell walls, by the way, but these, the ones we're going to talk about right now, do. And actually, the bacteria with the cell walls are the ones that are typically most resistant. The bacteria that don't have walls, they're very sensitive to this type of thing. Okay, because they're basically like a protist or something like that. They're just a cell sitting out there with membrane and things like that. Okay, So imagine we have, let's just for all of these, let's put 10, just arbitrarily, 10 units of salt. Or whatever. It could be, I know I just did too many there. Salt, sugar, whatever, they're all solutes. It could be particulate matter from like smoking or whatever, all that type of stuff. So let's take cell number 1, and these are all like in individual environments. I should be using different colors right now. Whatever. All right. So outside is water. It's an aqueous environment. But let's say in this first one, we've got solutes on the outside here. Let's say there's also a concentration of 10 out here. Okay. So that means the inside of the cell and the outside of the cells are the same. Now over here, let's say there's more stuff in there. We get the 10. Let's say there's only like 4 on the outside. So there's a greater concentration on the inside than on the outside. And finally, let's flip that around. Let's say there's like 24. I'm just kind of making up these numbers. But you can see there's much greater concentration outside the cell than inside the cell. Okay, so now which way is the water going? Now we'll use the blue. Let's do this. There we go. No, let's do a fancy pen. Blue, all right. Which way is the water going in this situation? Is it going in, going out, or is it not going anywhere? Well, in this situation, the water isn't moving anywhere. I mean, it is moving in, but it's also moving out at the same rate. So this is what you would consider isotonic, or the same. The net movement is the same, because water's freely going in and out. This is balanced. Now here, we have a higher concentration on the inside than on the outside. So remember, the water is what's moving. So the water is going inside. That's it. Water's going to flush in and try and dilute the inside of that cell. All right? This is called a hypotonic environment. We're talking about the environment in relation to the cell. Now you could flip it around. You could say the inside of the cell is hypertonic to the environment, but typically make sure you pay attention to the way these are worded, and usually they're talking about the environment in relation to the cell. So this is a hypotonic environment. So less solute on the outside, more inside, water flows in. Final one over here, it's the opposite. I just said the word already. This is hypertonic. In other words, the environment has more solutes than on the inside. So where's the water going? It's rushing out of the cell because it wants to dilute the world. Wants to save everything. It's kind of a neat pen, but it was a little bit of a mess. Right? Okay, so let's review that again. So this is isotonic. Net movement is 0. Okay, this is hypotonic environment. And that means the water is rushing in. And this is hyper with an E. This is a hypertonic environment. So what is going to happen? What is the fate of these things? Well, isotonic is fine. This is where those non-cell wall ones are going to live. This is perfectly happy. Maybe you won't have the high pressure. Plant cells don't like this. Plant cells want to be a little more rigid over here in this hypotonic thing. Some bacteria want to be a little more rigid too, but this is certainly not like a death sentence. Hypotonic, water flushing in. Now, without that wall, we could have a lysis. It could just burst, which actually can happen. It can happen in certain environments. It can happen to your own cells, all kinds of things. It does take a lot of water and stuff like that. But this is actually, plants kind of like this because this pushes the pressure of the cell membrane up against the cell wall. It keeps them nice and rigid. And a lot of times plants do enjoy this. This is why they get really weepy or wilty when there's not enough water and stuff like that. They do things for various molecular reasons. There's all kinds of pathways that need certain levels. But plants like the rigidity. Bacteria can handle it too as long as they've got that really nice rigid wall. So they'll be fine. But in this situation, now the wall actually works against them. In the hypertonic situation, the membrane is going to shrink and it's going to kind of shrivel up and it will rip away from the wall, causing it to lyse. And it will tear apart and we get death. Okay, so this situation, this is the one where we end up having cell lysis. Okay, cell death. I'm going to undo that. That was terrible. Let's use a better pen. Okay, so this is where we get the lysis. In other words, it's the rupturing of the cell in the hypertonic situation can rip apart. Net flow is outside, right? Plasma lysis. Great, okay? So that's what's kind of happening in this. Now, a couple things. Let's take a left turn here. This type of preservation, or this type of thinking, has gone into food preservation for a long, long time. It's been observed. People kind of knew that if you get some food, here's my terrible fish drawing. It's actually not as terrible as I thought it would be. Right? We catch a fish, pretty soon it goes rotten. We don't have a refrigerator. What are we going to do? Well, if we pour salt on it, right? Or maybe we smoke it. We put it on a fire and we smoke it. Or maybe we hang it up and we dry it. Well, we add to the solute concentration, right? We dry it out so the water, H2O, is out of there, right? Smoke it. We're adding to the particulate matter. Salting, we're adding to the particulate matter. What are we doing? So now, if a bacteria lands on that, remember, we're changing the environment, not changing the bacteria. So now here's a little bacteria cell. It lands on there. Hey, guys, what's up? I want to eat some of this fish. And it gets in there. And, oh, no, look at all that. Look at all that salt or smoke or whatever. Boom, there it is. Water rushes out. Plasma lysis. Okay, so for many, for thousands of years, salting, smoking, drying meats, things that now have to be refrigerated, well, those were used because they prevented microorganisms from living on there. You have other things like foods that are high in sugar, jellies, jams, like preserves, chocolate, things like that. They don't spoil the same way because they have so much solute concentration, things land on them and they die. Okay, so just the concentration of that stuff. I mean, humans nowadays, we eat so much sugar, maybe we'll eventually one day be resistant to bacteria. I'm totally kidding. But this is a true thing. This actually happens. So there's many cultures still today, salt and smoke things, dry things. It prevents the bacteria from being able to survive on there because of this osmotic pressure. That's how it all works. So here's what we would do with this lab. Well, we'd take some controls. We'd actually probably take our unknowns. We'd put them on some different plates. Put them on some BHI to see how well they grow. But we can't just have BHI on here. These are going to be BHI plus sodium chloride. So some NaCl. And you put different percentages on here. I think when we do this, we do 1, 5, 10, and 15. Typically, I think the book goes a little bit higher. I think the book goes all the way up to 25. But I think we just do 1, 5, 10, and 15, right? So 1% sodium chloride, 5% sodium chloride, 10% sodium chloride, 15% sodium chloride. And so what can live? Well, here, just about, we're doing pretty well. There are some bacteria that are halophiles where they have to have high amounts of salt. Okay, so the halophile, salt-loving or osmophiles, because we're talking about sugars is usually osmophile. Halophile is specifically salt. Some things have to grow on super high salt, but we're really not dealing with anything like that in this lab. But 1%, pretty much anything can live. 5%, well, you're going to see things like E. coli. It's not going to be able to live anymore, right? But if you have a Staphylococcus epidermidis or Staphylococcus aureus, they're going to do just fine. In fact, they're going to do fine here, and they can even sometimes survive up to 15%. Most of the time, the 15% is usually around their ceiling. And so, yeah, we can see this. So some bacteria, they're able to cope. There's different ways they can do it. They have these things that they can express, osmoregulators and stuff like that can help protect them. Transport of osmolytes and osmoprotectants. Like they can basically, they can kind of accumulate and transport for all these electrolytes and other things that help balance it out. So if they end up in a situation with a lot of salt, they have these mechanisms that kind of move some things around and it kind of boosts up their own salt content, so they're able to stay. So like the Staphylococcus can do that pretty well. Staph epidermidis, Staph aureus can do that. E. coli not so much. It's going to be pretty sensitive. I think sometimes we can get it on the 5%. That's usually about where it stops, because it doesn't have that capability. It's not equipped to live in an environment with that much salt. And so you see that and you think about this. So what are these applications, right? So we eat salty foods and there's salted foods out there. Well, there are bacteria that already exist that can sometimes handle some of that high salt levels. And there's halophiles that are even up higher. Some of those are up to 25% or so that can really, really grow in super high salt content. That would be so salty you probably wouldn't even want to be eating that stuff. So just keep that in mind. It's all about these growth curves. And there's a lot of stuff too. It's pretty neat. But osmotic pressure, certainly something that bacteria have to deal with, and some of them are really well equipped to handle, others not so much. All right, thank you guys.